Given that we wanted to create a reference genome, it had to be an individual that was ready to put it into a public domain situation, publish it to gene databases, publish it in the scientific literature. Now, Craig was clearly in a position where he was ready to do that. It's a very brave step, but it's a really important first step in order to generate a reference, to have an individual person's consent to use their genetic material.
I think that what is truly important here is starting also a process where it is clear that people can obtain their genome sequences, that it is not something to be fearful of, that in fact, in most cases, it's going to be information that will be interesting, but it will not immediately impact on that individual, but it will contribute to the greater health of human beings in the future.
So, well, there's always been a lot of concern from the beginning back at Celera, and one of the reasons I volunteered to have my genome sequenced was to show leadership in a field where everybody thought that it was a very high-risk event to have your genome sequenced. I have no difficulty having mine on the internet, not only because it's difficult to interpret, but I think it's because there are very few incidences in human genetics where there's a direct causal event in some absolute deterministic fashion between genetics and a trait. Almost everything we have is some sort of trade-off between genetics and the environment, and seeing the big picture, I think, is going to be important for all of us.
In 2001, and for the brief period of time it took us to essentially assemble a genome and put it together. At that time, it was 5 individuals. Essentially what we generated, and what the public consortium as well in their own fashion generated, was essentially a mixed sequence. So the best way to represent that was as a haploid sequence. Basically, a haploid sequence takes 2 chromosomal copies and merges them into one. What we've done today is essentially an improvement on that. Essentially, we've taken all the sequence from a single human, Craig Venter. We've essentially taken all the sequence reads, we've put them together into an assembly, and we've characterized not just the single base pair changes that differ, but all the variation.
The first thing in doing Craig Venter's genome was to just start to learn how to do a personal human genome. And we did this in a way where we used the very best technology. It wasn't necessarily the cheapest technology. We weren't looking to use the cheapest technology. We were looking to use the best technology that would allow us to develop the sequence of a diploid human genome, and one that would represent that individual's genetic content.
You really begin to understand the relation of genes to environment. You begin to understand from this the ability that your genome can respond to an environment and how it may respond. And again, this response is really described in terms of probability. There will be events that you can almost predict with great certainty will happen to an individual once you know their genome. We know that already. Huntington's disease is very clear. There are other diseases for which it's, even with many more years of research, we will never be certain to really be able to say, yes, you will have some aspect of cardiovascular disease in your life, or you will have cardiac arrest at 60. We will never be able to say that with certainty. What we will be able to say is that there is some probability. You have a 50% chance, you have a 70% chance that at some point in your life you may suffer from this condition. And knowing your genome and having your genome in your hand on a disk or on a database, in a database somewhere, will enable you to then go to the biological knowledge, the biological literature that we're accumulating in many labs around the world, and essentially put together a profile of your disease risk. At the Venter Institute, we've been able essentially to put together a methodology on how to assemble, identify the variants, and build a diploid sequence from a human. Once you can do that, you can do it many hundreds and thousands of times routinely. And really, our work has been an aspect that's technical, has really essentially been to define the methodology. What are the caveats? What are the issues once you start doing this? With more automation, faster sequencing, better databases, we'll be able to essentially accumulate this for hundreds of individuals, thousands.
Well, whether everybody wants to actually read all 6 billion letters of their genetic code, that's unlikely. I think there'll be probably a new industry set up around the complex interpretation of the genetic code, where people will probably get reports from their genomes. But for those that are slightly more curious, it's actually an exciting endeavor. I'm the first one on the planet that's had that privilege to do that, but each time there's a new scientific study published about genetic relationship with traits or disease, myself or someone on the team can go in and look at my own genetic code and look at those variants and try and understand these traits in terms of own knowledge, family history, etc. And I think we'll be in that era a long time, where the curious members of the public that have their genome sequenced will probably want to do this, and there'll probably be a lot of new tools to enable them to do that. Others will just rely on interpretation and the reports given to them. But I think it will start the era of genetically based preventive medicine. At least where we know there's genetic components, we should be able to alter lifestyle, take preventative drugs like the statins, for example, for cardiovascular disease, that have a chance of lowering the risk for disease going forward. As we understand the basis of disease, there will be more and more incidents where that would be possible.
Diseases like cancer, cardiovascular disease, obesity, these are phenotypes that result from the effect of many different genes. And until we start taking that approach of understanding that we need to know the complete genomic constitution of an individual, and we need to have information about their medical history and be able to follow them as they develop as an individual. Until we have that, we truly won't have the era of personalized genomic medicine, and we won't see the fruitions of the Human Genome Project.
So clearly, we have Craig's genome. We also have James Watson's genome, and those two individuals provide us essentially with a pioneering moment, if you like, to understand the inter-individual variation. So clearly, for us, an interesting first step would be to compare Craig's genome to James Watson's genome. I think this is a very interesting scientific endeavor, because it essentially will tell us what we can expect when you compare two people once you know their full variant set. That's the most interesting aspect, a first aspect of personalized genomics.
So now the job of explaining human-to-human variation is going to be a much bigger challenge across the population. Between two individuals of dissimilar backgrounds, different ethnogeographic locations, for example, the variation might be as high as 1% between their chromosomes. So I think this is going to have a very significant impact on our thinking. But we're about to enter the era of sequencing large numbers of human genomes. This is the first diploid genome and provides a different comparison source than the haploid genomes that Celera published before and the federal government published before. So this is beginning a new era of absolutely individual genomes, having a unique basis of comparing them with all the variation. And I think it's going to be a very exciting era going forward.
It's not sufficient just to identify DNA variation. We need to know the relationship between each variant. Know for each person the set of signposts that define their chromosomes, basically. Once we have that, then we can truly begin to do disease studies. We truly can, rather than hunt for the regions of somebody's chromosomes that are implicated in a disease, we can essentially go straight to the genes.
I think people now understand that this is just the beginning, and we do need these thousands. We need millions of human genomes in order to truly do for humankind what we'd like and to make everybody's health situation better than it is today.
That large database will allow us to basically answer any question about what's genetic versus what's environmental. The nature-nurture questions, it'll probably be a different answer for every trait, every condition, every disease. Some things will be entirely genetic and other things will be almost entirely environmental. Most things will have components of both, but that's the only way we will understand the wide variation.
And this, what we're essentially doing here is empowering medicine to understand more. We're essentially providing them with a fine-scale test that goes beyond blood pressure maintenance. It goes beyond doing a blood screen or any other kind of test you can have. It supplements those tests and it provides them with a lot more information. And clearly this is our place. This is our place as scientists. We can essentially empower medicine. That's our hope.
And having complete genomes gives us the ability to begin to unravel these mysteries of human evolution, human biology, and even maybe predict the future.